that works. So, right, remember we're going backwards, right? The light dependent technically happens first. It's all a big cycle, but you have to have the light dependent first before you can have the light independent. But we're going to start with the light independent because it's perhaps a little trickier. So this is available, right? The photosynthesis study guide is available right under our photosynthesis quiz assignment. So you can access that, start studying for that. That will be a week from today, right? So next Saturday. That is also the end of the quarter, so make sure that you check in on your portal and see what's going on for grades. The lab report will be included in third quarter and this quiz, so those are two big assignments, so chances are your grade could change dramatically from what it is right now, right? So that could be an awesome thing for you, right? It could go way up, or it could go way down, so keep that in mind. All right, so in terms of where we are for today, right, we're going to put the light-dependent reactions on hold for now. A little more complex than the light-independent reactions, right? For the light-independent reactions, they are happening in the stroma, right? So in that sort of fluid that's in the center of the chloroplast, but kind of like the cytoplasm. For the general ins and outs, right, sort of the overall reaction that we're looking at here, right, is we've got, so we're in the light independent reactions, LIR. This is sometimes called the Calvin cycle, in part because it was discovered by this guy named Calvin, right, so they named it after him, Calvin cycle. And generally what happens here is you take in some carbon dioxide gas. How does this get into the leaf? Yeah, through the stoma, the stomata, the pores in the bottom of the leaf. You take in some CO2 gas. You add some electrons from our electron carrier, NAD, DPH, right? And you add a little energy from ATP. So those are our reactants for the Calvin cycle. You get out, right, the products of this reaction are going to be sugars. That's unfortunate. Sugar, right, in some wonky language, and NADP plus, wow, this thing needs to be recalibrated, shoot, plus ADP, wow, that says ADP, in case you're wondering. I feel like the top half of the board, like from here to here, is well calibrated, but this is just not.
Wow.
That's pretty funny. Okay, right, so this is generally what we're looking at, and this happens in a cycle. So, right, here we are, light-independent reactions. The reason it's called the Calvin cycle, right, is that it's, you know, the ends in some way feed back to the beginning, right, so that it can restart again. I don't need you to know there are technically 3 stages of the Calvin cycle, carbon fixation, reduction, and regeneration. I don't need you to know that. What I need you to know, right, is that this cycle, what happens is you take in CO2 gas, you add an electron, you add some energy, and you get this sugar molecule, right? G3P is what it's called. Also don't need to know that, but this is a precursor to glucose, right, which is this sort of magical sugar molecule that makes a lot of ATP. One molecule of glucose can make 36 ATP, which is a lot of energy. So if we can create one glucose molecule from just a little bit of ATP, that's a net positive, which is why the plant does this. What's up, Charlie? Yeah. So Calvin cycle, right? CO2 turns into sugars. This is catalyzed, this series of reactions that creates this by an enzyme. The enzyme is called ribisco, okay? And ribisco is actually a acronym. It stands for ribulose 1,5-bisphosphate carboxylase oxygenase. You do not need to know that. You just need to know rubisco. Okay, but this is sort of a magical enzyme. It's really important, and I think it's the most abundant enzyme on Earth, right? Because every single plant needs rubisco to undergo this Calvin cycle, which is really, really important. However, despite being so abundant and so important, rubisco is an imperfect enzyme, okay? And the reason that it's imperfect is because of this carboxylase and slash oxygenase, right? The thing that comes before the A's here in the way that it's written. Okay, chill out. My smart board is a dumb board, yeah. Right, the word that comes before the A's, what does the A's mean here? What does it indicate? It's an enzyme. And typically the word before the A's is the substrate that it's acting on, right? The thing that it's reacting with. And typically most enzymes only have one thing that they react with, right? They're specific. That's the definition of an enzyme. But rubisco has this weird thing where under certain conditions, it can react with oxygen. It's supposed to react with carbon. In an ideal world, it's a carboxylase, right? It reacts with carbon. But in some conditions, it will accidentally bind to oxygen and react with oxygen, which is a problem for the plant. You can see here from this picture, right, that when the enzyme binds with carbon, right, carbon dioxide, it goes through photosynthesis and creates energy, which is awesome. The plant wants to do that, right? It wants to make energy. But under certain conditions, when the rubisco binds to oxygen, it undergoes this process called photorespiration, which is basically bleh. It makes waste. It doesn't make anything. So when a rubisco binds to an oxygen, that is bad for a plant, right? It is wasted time, wasted energy, and it makes nothing. Makes bupkis for the plant. So one of the things that can increase the chances that rubisco binds to oxygen instead of carbon dioxide is actually, so we're looking at rubisco, and it will bind to oxygen more often at high temperatures. Okay, and this is bad, right? The plant does not want this, right? You know, at regular temperatures, right? Most plants that live around New Hampshire, you know, they will undergo some photorespiration, but it doesn't get so hot here to disrupt the enzyme quite as much. So in a plant, say, you know, a plant in, you know, New Hampshire has way more rubisco, but say it has, you know, 5 rubisco working at a time, and in one instance, one of those rubisco binds to oxygen, right? Is that a huge problem for the plant? No, I mean, like, it's not ideal, but it's still got 4 functioning rubisco. What would happen if we took that plant, right, if I took a, you know, plant native to New Hampshire, like a fern or a maple tree, right? If I took that tree and tried to plant it in the Mojave Desert, right, where it gets really hot, what would then happen is maybe, you know, 4 out of the 5, 5 out of the 6 rubisco would start binding to oxygen, right? Now all of these rubisco are making waste product, not energy, right? Yeah, so the plant would not be efficient in creating energy and creating glucose, and it would eventually just die. So part of the reason, actually, can anybody think why high temperatures might make it more likely that rubisco binds to oxygen, its unintended substrate? Jack?
Would like the oxygen be like a bigger molecule, so like expand through the heat?
Good thought. Oxygen is actually smaller than carbon dioxide, so it's not going to necessarily expand. But what do we know happens to enzymes when they're heated? they denature. And what is denaturing?
Like unravels, basically.
Yeah, it kind of unfolds. The structure becomes less defined.
So
Right.
you fit a different substrate.
Exactly, right? So the heats, right, that we're talking about here aren't high enough to denature Rubisco, but they are high enough to start those molecules moving a little bit, right, making the active site less rigid, right, so that it can kind of, shift around and make oxygen work even though it shouldn't work, right? So this is where the enzyme piece that we just learned ties in here. Now, obviously, if I took a maple tree and I planted it in the Mojave Desert, it would not survive. But there are plants that live in the Mojave Desert and do fine, right? Those plants have the same enzyme, the same Rubisco that maple trees have, but they have other adaptations that allow them to live in high heats and keep the Rubisco working with carbon dioxide rather than oxygen.
Does it keep Rubisco cold or something?
Not exactly. What these plants do is they kind of, through different mechanisms, which we'll talk about in a second, they sort of separate their Rubisco from their gas intake, right? So when the leaf opens its stomata, CO2 and oxygen come in, right? The plant wants to ensure that the Rubisco is only exposed to CO2, or at least a higher concentration of CO2 than oxygen, right? So if it's selectively binding to oxygen because it's hot, we just want to give it CO2, so it has no option to bind to oxygen. Does that make sense? So there are two types of plants that do this, and they are called CAM plants and C4 plants. The plants that we think of that live in New Hampshire are denoted as C3 plants. Cross the magic line on the smartboard. So C3 plants are they're regular plants. They don't do anything special to prevent photorespiration in their Rubisco, right? They're just, they live in pretty temperate climates, so they can just send both the gases in and not really care about it. CAM plants separate their... So CAM plants are going to separate their Rubisco from gas intake by time, and we'll talk about what I mean by that. So they're going to make sure that their Rubisco is only subject to CO2 by manipulating the time that those two are coming together. And C4 are going to separate them by space. Okay? So they're going to physically have the Rubisco in a different place from the gas intake so that they can only send CO2 towards the Rubisco. Does that make sense? Examples of CAM plants are the plants that you think of living in really, really extreme environments. That's like cacti, succulents, some like desert grasses, beach grass, things like that. C4 plants are actually really efficient in making sugar. They make a surplus of sugar because of this design of separating their Rubisco by space. So can you think of any plants that make a lot of sugar? Yeah, so like really sweet fruits. Think like above apples. So like mangoes, pineapple trees, sugar cane are all C4 plants, right? They're making just like a butt ton of sugar, right? So let me throw those up there. So we've got cacti, beach grass for CAM plants, and then C4 are any sort of sugary plants that you can think of. Pineapple, sugar cane. Okay. So let's try to wrap our head around how a plant would separate these two processes by time and space. That's not what I wanted. There we go. So CAM plants stands for Crassulian acid metabolism. You don't have to know that, just know CAM plants. But if you were curious, that's what it stands for. And this refers to the fact that they store their carbon dioxide as an acid. So what these plants do, and the main reason they do this is because they live in really hot environments. So their want to separate these processes by time is twofold. Because if they do their gas intake during the day, right, if they open their stomata during the day, what happens? What do they lose in the high heat?
A bunch of water.
Water, right? Water will evaporate out. So they try their best to keep their stomata closed during the day, right? And then they'll open them at night so that when it's cooler, they're not going to lose a lot of water via evaporation, right? This also helps them keep their rubisco saturated in CO2. So what they'll do is at night, they'll open these stomata, right, and the CO2 will enter. Along with the CO2, there will be some oxygen that comes in, right? They turn the CO2, you don't have to know these malate and malic acids. I don't care about that. Just know that they turn their CO2 into another substance that then can be stored in a vacuole, right? So they take their CO2, they bunch it together, turn it into another substance, store it in a vacuole during the night, close their stomata during the day, right? And then they turn this substance from the vacuole back into CO2, right? So by doing that, right, and keeping their stomata closed during the day, they're decreasing the amount of oxygen in the chloroplast and increasing the amount of CO2, right? So this is twofold for cam plants, right? They are saturating their rubisco in CO2 and also preventing water loss by keeping their stomata closed. So that's part of the reason that these cam plants, these desert plants, will separate these processes by time. Does anyone have questions on that? Okay, this is a confusing, not confusing, but it's got some big words in it, this picture. Just know, right, we've got CO2 coming in, it gets turned into something else that's really easily stored, right? Malic acid has 4 or 5 carbons in one molecule, whereas CO2 only has 1, right? So this one malic acid molecule is the same as 4 CO2s, right? So it's kind of like, and it's smaller, it's like super powered, right? So then when you release it, you have 4 CO2s. So it's kind of bunching it together and then sending it back out. Does that make sense? Does anyone have questions on that? So this is CAM, all right? C4, our sugary plants, is slightly different. So remember, the C4 plants are going to separate their gas intake from their rubisco by space, sorry, right? So what happens here is the leaf of a a C4 plant is going to have 2 different layers of cells. They've got a top cell, so this is the top cell here. This is the top part of this here is the external part of the leaf, right, where the stomata is located. You can see the gas is going to come in through here, right? So the stomata are open during the day, they're open during the night, right? Gas is coming in and out. But what happens is this upper cell, there are no rubisco in this upper cell. There are no chloroplasts. So there's not going to be any photosynthesis going on in this upper cell. There are rubisco in this bundle sheath cell that's right below this upper level of the cell, right? So the CO2 gas comes in along with oxygen, and although this layer of the cell is permeable to gas, right, because of the stomata, this middle membrane here is not permeable, so the gas can't pass through these two from one cell to another, right? In order for the CO2 to get into the bottom cell where there is rubisco for it to do photosynthesis with, it needs to get turned into something else. So in this top cell, right, we're going to turn the CO2 into malate, which is a carbon molecule. It's a 4-carbon molecule, hence why this is called the C4 pathway. This malate then can be shuttled across the membrane, right? The membrane's permeable to malate, right? So the malate can move across the membrane, where it then gets turned back into CO2, right, and can be fed into rubisco, right? So these plants are super efficient because no oxygen passes the membrane into rubisco. C3 plants, right, which are the plants that we think of around New Hampshire, right, are going to do a little bit of photorespiration. They're not going to be super efficient at making sugars. These C4 plants are going to be really efficient at making sugars, right? Because they are doing zero photorespiration. There is no oxygen that can come into contact with this rubisco. Okay. In terms of what you need to know here, just know that they're separated by space. I'm not concerned that you know that it gets turned into oxaloacetate and then malate. Just know that it gets turned into something else, right? Crosses the membrane, gets turned back into carbon dioxide. Okay? So just an overview here, right, of the 3 different types of plants that we've touched on. C3 plants, right, these are the plants that we think of in New Hampshire. They're not doing anything special. They're allowing gases in. Both CO2 and oxygen can interact with rubisco, but because it's at a reasonable temperature, more often than not, they're going to bind CO2, not a huge issue. C4 plants, right, sugary plants that need to make a lot of sugar, they're going to separate the 2 processes by space, right? And CAM plants, which need to conserve water and also protect their rubisco from photorespiration because they live in really hot environments, are going to separate those 2 processes by time. Bella?
So they take in CO2 during the night and then have their Calvin cycle going during the day?
Exactly. And we'll talk a little bit why most of these plants are only going to have their Calvin cycle can go on a little bit at night, but most of the time they're not doing the Calvin cycle at night because they run out of ATP and NADPH quickly. Where are ATP and NADPH made? The light-dependent reactions, right? So even though we call the Calvin cycle light-independent, it is sort of light-dependent because it needs the products of the light-dependent reaction to run. Does that make sense? Cool. So this is, I think, chapter 8.3 in your book goes into this. And this is sort of the overview of the light-independent reactions. What we'll do the next time we'll meet is we'll go over the light-dependent reactions, which are a little harder to get your head around. So for homework this weekend, I'm having you read about those light-dependent reactions. It's like, I assigned like a page or 2 of reading in your textbook, just like one little section. So hopefully you'll get to spend some time with that, wrap your head around it, so when we come back together, you're a little ahead of the curve. All right? What? And for bonus, watch Bill Nye, I guess. So your study guide is up on the portal, so you should have access to that if you need it.
Wait, this is why don't plants even grow fruit?
Um,
They don't use